Hello, everyone, and welcome to this session of the Cognitive webinar. I'm Yunus Şahin from CogIST, and I'll be the moderator today. We are quite excited to have Michael Anderson from Western University here with us, who works on, broadly speaking, the philosophy of neuroscience and philosophy of cognitive science. Many of you will be familiar with his influential work After Phrenology, where he criticizes the traditional approaches to localization and functional neuroimaging, and in fact, criticizes traditional functional localizationist approaches in general. And in a similar vein, today's talk, replacing functional localization with psycho-neural-behavioral constraint, introduces an exciting line of recent work, novel work. Over the past few years, Anderson and his team have been developing alternatives to traditional GLM-style localization, exploring instead dynamic in relationships between perceptual information, behavioral patterns, and neural activity altogether. So today we'll talk about these studies and the framework and consider broader implications for the field. Before we begin, a few practical notes. This session is being recorded and will later be available on our YouTube channel. So if you're not okay with being visible on the recording, so let us know. We can edit the recording before uploading it on YouTube. If you feel comfortable, we encourage you to keep your camera on. It helps create more interactive and maybe natural setting, natural atmosphere. Please keep your microphone muted during the talk. We will have a Q&A session after the talk. And during that Q&A session, you can raise your hand via the button below and ask your question out loud, or you can type your question in the chat, however it suits you. Michael, thank you again for accepting our invitation and being here today with us. I think we are ready and we can begin whenever you say so.
Okay, let me hit the screen share. Thanks for the invitation. I'm looking forward to it and the discussion. Okay, that's visible?
Yeah,
And
everything's great.
Perfect. So as you just said, I will be talking about some brand new work in which we're trying to provide scientific alternatives to the kind of normal way of going about doing functional neuroimaging. In order to get there, I will be going over some older work criticizing functional localization, basically just to motivate what it is that we've been up to in my lab for the last couple of years. So this is the talk title I shared, Replacing Functional Localization with Psycho-Neural-Behavioral Constraint. I thought an equally and much more fun title would be this one, In Praise of Ecological Thruples. So evidence for psychoneural behavioral coupling. So there are various issues, I think, that are worth appreciating in functional architecture of the brain. One is neural reuse, which you just mentioned. That's the topic of After Phrenology. I'll go over some of that in detail. There's also ubiquitous neuromodulation. That is, individual bits of the brain appear to change their function and certainly change their connectivity under different circumstances. And that is not fully appreciated, at least in large-scale human neuroscience, although it's much more appreciated in, say, invertebrate neuroscience. There is, as a result of these things, functional complexity at multiple spatial scales in the central nervous system. And one thing I'll just mention, but we'll need to come back to this at the very end. The other issue is that the brain is a very causally dense system, by which I mean that its parts interact in a highly robust way. And so parceling it out into individual bits for which you can identify the function is an inherently difficult task, extremely difficult, maybe impossible. So for all these kinds of reasons, I spent more than a decade now thinking about what's the issue, what's the real issue, what's the problem with this attitude and this way of doing science. But also, as I'll show you some of today, trying to come up with viable alternatives, which is, of course, vital if you're going to make progress. Okay, so I'll just give you some background on neural reuse. I hope, well, I kind of hope you are familiar with it, of course, but I'm assuming you aren't very familiar, and I'm going to go over just a few of the real basics of neural reuse and what it implies for functional architecture. And I'm really just going to hit the last two of these 3 points. So the first is that the reuse of individual parts of the brain across multiple tasks you can actually see in the evidence. And the other bit I'll give you a bit of evidence for is that the differential function, so differential behavioral and cognitive function, isn't the result of differentially activating different parts of the brain. It's instead the result of dynamically combining the same elements in different coalitions. So again, I'm going to go over just a few data points to establish these two things, then we'll move on to the newer stuff. So a strategy here is what's called a functional diversity analysis. It's not that difficult a thing to do. First, you build a large collection. Right now we have 6,300 experiments of brain imaging data. And you categorize the cognitive processes that are being probed in each of the experiments: emotion, visual perception, auditory perception, or more fine grain. We've done it at various levels of grain. And then you calculate the diversity of function that's observed in each part of the brain. Well, how do you do that? Well, we borrow something from ecology. Shannon entropy you're probably familiar with in multiple contexts, but it's used in ecology to calculate species diversity in an ecosystem, right? So you want to know how diverse an ecosystem is, whether diversity is going up or down. Say, if you're interested in ecology, you're probably interested in things like the rainforest and what's happening there, given encroachments by agriculture and global climate change. This is an approach, and you can say, diversity is going down, which it is, in fact. And this gives you a way to actually measure it. Okay, so basically, we just did a simple analogy. So in ecology, you say, well, if you have a region with many different plants and animals from many different species, that's a higher diversity. So close to 1, we scale it from 0 to 1 just to make it easier to see. And then if instead most plants and animals you observe in a particular region of your ecosystem come from a single or just a few species, well, that's going to be a low diversity region, so closer to 0. Well, here's the obvious analogy. If you take a region of the brain in which there are many cognitive functions from many different categories of function, well, that's going to be a high diversity region. If instead most cognitive functions are in a single or a few category, that's going to be a low diversity region, right? And this is the classic way of thinking about it, where vision is localized back here and language is localized here. Well, so we did this. The data point I'll show you a few, but the first one, we just took a 10 millimeter sphere drawn around every voxel in the brain, right? And a voxel, as I'm presuming you know, is a volumetric pixel, so it's just what we use in neuroimaging to pick up a spot to look. So we drew a 10 millimeter sphere around every voxel in the brain, and we just did this calculation. We looked at how many cognitive functions were inside that sphere, and whether they were in many or a few categories using Shannon entropy. Well, here are the results. You can see it's massively skewed towards the upper end, right? I'm going to put a few regions on here. So this 0.88 is a voxel in left anterior insula. And you can see this is just 11 categories. I'll show you a version of this with 20 some odd different categories. Anyway, 11 categories. You can see that voxel in left anterior insula is active in pretty much all 11 of the categories we looked at. Less so in category 11, which is vision, much less so, in fact, but very highly active across many other categories. So it ends up with a diversity of 0.88, so quite close to 1. Now you'll see from the histogram where 0.88 is. So that's very high diversity, and not a lot of the brain is that diverse. Okay. In contrast, look at a voxel from left auditory cortex, the 0.41. It's almost entirely active in audition. So that's a relative specialist. It is a little bit active in action tasks and in language tasks. Um, uh, but again, looking at histogram where 0.41 falls, almost nothing in the brain is that dedicated. There are a few specialists, and as you can see, if you can see the tail going down there, there are some that are, basically zero diversity. That only does one thing, at least in the data set we have. But most of the brain is not like that at all. So that brings us to this voxel in the left thalamus. That's the 0.76. 0.76 is the median value for this histogram, right? So the median part of the brain has a diversity of 0.76, which means, as you can see from the spider plot, that is active across quite a number of tasks. In this particular case, it's active across a somesthesis, which of course, this is the textbook version of thalamus, what it's up to. But it's also active in action tasks. It's active in attention tasks. It's active in reasoning tasks, and so on, right? Now, an important point to make here is that none of the data that I'm showing today, and none of the arguments that I make across the work on this topic suggests that there's not functional differentiation in the brain. As you can see just from looking at these spider plots, different parts of the brain have a different mix of functions, right? We just illustrate that even more so with this. This is a much more fine-grained analysis. And these are the 6 divisions of the insula. So dorsal anterior insula, posterior insula, ventral anterior insula, left and right. These are neighboring structures in the brain, but if you look at these spider plots, they're really quite different from one another, right? So one of the things, and this is kind of a philosophy of science point or philosophy of neuroscience point, we inferred functional specialization from our observation of functional differentiation. That turns out to be a bad inference. There's definitely overwhelmingly functional differentiation across the brain, but there does not look like there's very much functional specialization. And again, that makes identifying localizing function to individual pieces of the brain, a highly uncertain task. I can't say impossible because all the data aren't in and they never will be, but it's highly uncertain. And so it means we probably want alternatives. Let me turn now to the connectivity analysis, right? And this is the claim that differential function is the result of dynamically combining the same elements in different coalitions. Well, here we do, again, this is a functional connectivity analysis. So we ask, do parts of the brain form different partnerships for different functions? And this is basically a chi-squared test for those into stats. But you're basically looking for evidence of statistical dependence in the activity of different brain regions. In other words, if we have activity in region A, does that make activity in region B more likely and vice versa? Okay. So I just did a functional, same database, by the way. So I just, so here, okay, let me orient you now. This is the brain. This is the Harvard-Oxford Atlas rendered as spheres. And you can kind of see we're looking at the brain from the top, with the front of the brain at the top of the screen and the back of the brain at the back of the screen. And each of these spheres is at the geographical center of one of the 118 Harvard-Oxford anatomical regions. Okay, it's really just a convenient and attractive, I think, way of putting up the networks that I will show you, which are the functional connections under different circumstances. So here's the first one. This is just highlighting, well, you can kind of see in the background the full network, and it's quite dense. But I'm just highlighting for my purposes here, the partners are left precentral gyrus during semantics tasks. So tasks having to do with linguistic meaning, right? So it's got quite a number of partners, not the whole brain, of course. Right? Not everything is connected to everything. That's the other thing. There's functional differentiation. There's also specificity of causal connectivity, right? And that's important to the brain's function too. But here again, the specificity of causal connectivity shouldn't have given us the idea that there are modules, at least not in the traditional sense of a module. There are modules in a network science sense, but that's not a functional definition of modules. We can talk about that more if people are interested in that point. Anyway, here's left precentral gyrus in semantics tasks. That's left precentral gyrus in emotion tasks. That's left precentral gyrus in attention tasks. All right, let me just go back over that just so you can see the point here. And again, this is just visual inspection. You can also quantify this. I'm not showing you those data this afternoon. You can also quantify this, and it's overwhelmingly the case that there are different partnerships under different circumstances. Anyway, so here's semantics tasks, here's emotion tasks, and here's attention tasks, right? A different fan of connections for under every circumstance. And again, you can do this with much more fine-grained cognitive categories. This is just an easy way to make the point for this afternoon. So this is an example of large-scale neuromodulation. And there is compelling evidence for ubiquitous neuromodulation at all spatial scales, from genetics all the way up to this regional networks that I'm showing you, right? So this is in one of those invertebrates that I mentioned, right? This is C. elegans, the soil nematode. And each of these triangles is a neuron in its 384 neuron central nervous system. And so just look at the red one, ASH. ASH participates in all of the behaviors listed here. So aerotaxis, that is looking for oxygen. Octanol avoidance, it's a substance in their environment that they don't like. Aggregation, which is a social behavior, and repulsion, which is an antisocial behavior. The neuron ASH is involved in every single one of these behaviors. But in each of these behaviors, it has different set of neural partners. And by the way, it's governed by different genetic expression. This is another fascinating story I won't go into. Right? So even in an animal as simple as the soil and as ancient as C. elegans, we see this same pattern of individual neural elements being used across multiple behavioral circumstances and having that differential behavior be caused by not the differential activation of a given neuron, right? ASH is not solely responsible for any behavior, but by partnering with different neurons under different circumstances. That suggests this is an evolutionarily very ancient and widespread strategy for using scarce neural resources to create diversity of behavior. Okay, so that's enough for the kind of background information as to why you might be motivated to look for alternatives to functional localization, for the kind of one-to-one mapping that is just, for many people, still the gold standard of understanding how the brain works. I suspect that that's never going to happen. There has to be radical revolution in how we think about cognition, frankly, for that to be possible. But there are some other issues I won't go into in any detail, although again, happy to elaborate on these in the Q&A. I think there are really important issues with the stimulus response paradigm. And this is the overwhelmingly most common way of probing the brain, where you just provide a stimulus and you get a response. But behavior is not like that, right? Behavior is an ongoing interaction with the world, wherein there aren't stimuli. I'm in the middle of finishing a new book, and I have a whole chapter on how the stimulus idea is a very dumb idea. It came out of physiology, so it has a noble history, but it came out of physiology as the electric stimulus being applied to a muscle, right? And then suddenly became the thing you show people in psychology experiments, which aren't very stimulus-like, frankly. Anyway, that's the paradigm is problematic for a number of reasons. Partly for that reason, and partly, especially in neuroimaging, because you can't get much behavior inside, say, an fMRI. Most of the neuroimaging experiments are not ecologically valid. So that's a really important thing to try to overcome. And there have been various attempts to do it, right? So people watching movies in scanners, at least that's ongoing, dynamic and has some advantages for that. People doing the social, what they call hyperscanning, so interacting with one another in different scanners. So there are attempts to overcome it, but fundamentally, it's a problematic situation where you're lying on your back, being shown stimuli, and giving only simple responses. So that's a problem. I'm not going to solve all these problems, by the way, this afternoon, but these are the kind of things that worry me, and these are the kind of things that my group thinks about a lot. And then there are large questions about the applicability of the computational or representational theory of mind. In general, I mean that theory, which is still the dominant theory, has come under a lot of attack from multiple directions. So there are all sorts of reasons to be thinking hard about different ways of doing things in neuroimaging. So what I want to suggest is to gather all the issues I've mentioned above and the data that I showed you earlier suggests we need alternatives to methods that are rooted in passive viewing paradigms, to methods that are designed to specify function, especially local function. I guess that's enough for that. That's all I had in that slide. All right, so we need alternatives to these things, and these things are pervasive across neuroimaging. Okay. Now, let me then switch gears to starting to talk about the conceptual framework that I'm working with here. And one of the central concepts in that framework is something called resonance, neural resonance. Now, this is an infamous metaphor. Gibson was roundly criticized for invoking this metaphor. People are thinking, what do you think? The brain is a radio or something that resonates to waves? Anyway, here's just what he says about it. He says, in the case of persisting things, I suggest the perceptual system simply extracts the informational invariance from the flowing array. It resonates to the invariant structure or is attuned to it. And people aren't wrong to call this a radio metaphor. It is. Right? But what he's really referring to is the notion that rather than think about a stimulus being represented in some bit of the brain and then worked on by computational processes, instead what the brain does is couple to the flowing array of informational invariance. And this coupling is what he calls resonance. Now, this remained a largely criticized, roundly criticized concept, and nobody did much with it until my former postdoc, Vicente Raja, operationalized resonance in his dissertation back in 2018, and then he's published a few things since. The Mind in Machines, I think, is the... No, I think 2017 was his dissertation. 2018 is a Minds in Machines article, and 2021 is, I forget now which journal. I have the references at the end of the talk. Anyway, so he's operationalized resonance in a scientifically respectable way. And it's a really simple idea, a really nice idea. And this became the basis of the work that I'm going to be telling you about in a few minutes. So the basic idea is that organism-environment interactions are constrained by information available at the organism-environment scale, right? What's normally referred to as psi, or the Greek letter psi. The neural dynamics, chi, have their own informational constraints, right? How neurons respond to the world is constrained by the kind of information that they are exposed to, and of course the behavioral task that the organism is undergoing. So resonance then is just when both dynamics are jointly constrained by the same information. And that can be expressed, you can do it in lots of different ways. The simplest way is this linear equation, where chi is equal to psi times some coefficient, right? That's the one we'll be working with. Again, the brain is a nonlinear system, and there are more complex ways to express the coupling in a nonlinear way. We're sticking with the linear, in part because if we can show that there's linear coupling between information in the environment and neural dynamics, that's the hardest case to make. Right? The nonlinear case, because the degrees of freedom are so much greater, the nonlinear case is actually easier to make. So we're making the playing field difficult for ourselves so we can get a proof of concept out there in hopes that we can attract people to continue to investigate this idea. Okay, so in particular, we're going to be working with something called tau. Tau is something that's been extensively investigated in ecological psychology, and it's a measure of optical expansion that indexes time to contact. Right, so everyone will know as something comes towards you, somebody tosses you a ball, the apparent size, that's really a crude way to talk about it, but the apparent size of the thing will grow. In other words, the shadow, as it were, of the image that it projects on your retina will increase. And the rate of that optical expansion tells you when it's going to hit you or when you have to reach out to catch it. So this tau has been generalized to gap closings across multiple domains. So not just catching a ball, but doing even things like walking, knowing when your feet are going to hit the ground and things like this. Anyway, it's calculated as a value, whether it's visual angle, position, voltage, divided by its first derivative. Right? Another very simple mathematical concept. So tau is c divided by dot c. Okay, as I mentioned already, it's a very common ecological variable. Gannets, a particular seabird, gannets use it to time their wing folding when they enter the water when chasing prey. People use it to figure out when to hit or catch a moving ball. People also use it to know when it is or how it is they can approach objects. Birds also use it in less kinetic motions, so they use it to know when to start a perching behavior, things like this. So it's got a very long history, and it's well established as being one of the most important ecological variables. It's also been used a little bit in looking for neural resonance. So this is a van der Wiel and van der Meer article from 2009, and they did EEG with infants, basically just showing a looming target. And they discovered indeed evidence for coupling between the tau, that is the rate at which that target loomed, and activity in primary visual cortex. We liked this because it was a nice proof of concept, but for all the reasons I mentioned above, we have a non-behaving animal, the baby, not really even responding to the stimuli, just looking at it. And then their analysis of the neural data was very strange. If people know anything about EEG analysis, the coupling they looked for was using a single dipole model of the ERP. And then they looked for evidence of coupling in the single dipole ERP, which it's several layers away from reality. So I'm not sure why they made that choice. But it's an interesting article to read, and it was what was one of the inspirations for the work that we did. And so I do want to emphasize this is joint work with Vicente Iraja, my former postdoc. He's now a professor in Spain, University of Murcia, and Binqi Zhang, who was my undergraduate advisee and is now a master's student at Western. Anyway, here is the setup. Is there a pointer? So this is the setup. So it's virtual reality with the virtual reality goggles. We have a 64 channel EEG. And then the task is just catching a ball. Well, it's a block. I don't know why we rendered it as a block, but we did. So it's just catching a moving target, right? So it's a highly ecologically valid task. Now we used VR because we have full then control over the environment. Future work will do things like mess with the information visually, but we wanted to see whether we could even do it. We use EEG, but of course we need this person to be mobile and behaving. You couldn't do this task in any easy way inside a scanner, although I'll talk about something we did do inside a scanner, which is have people play Pac-Man, which at least has some of the dynamics you want in sort of a full behavioral task. That just turned out to be methodologically really hard to do. Anyway, so this is the goal. You start with a fixation cross, a target appears. Your job is just to catch this block before it hits or just as it hits that target, right? So a simple task. Okay. All right, so I mentioned a 64 channel EEG. We actually had three conditions. This has two because we had a fast condition where the intercept happened about 800 milliseconds after the onset of motion, a medium condition where intercept happened about a second and a half after the onset of motion. And then we had a slow condition, which we didn't analyze because it was so slow that people just watched the thing and then reached out to touch it at the very end. So it's not the same task at all. So we're ignoring the slows. We're just looking at the fast and medium conditions. And then here is just the map of the 64 channel cap. All right, so each of these is an electrode. Again, I'm assuming you're mostly familiar with EEG, at least in general, but you just place electrodes all along the scalp. Those electrodes are measuring electrical activity at the surface of the brain. And of course, that activity indexes the summed activity of all the electrical activities happening in the brain beneath it. Okay? This is just a standard 64 channel cap. So we had 17 participants, 100 trials per participant per condition. We did standard EEG data pre-processing, so all the filtering and stuff we need to do to get rid of noise and all that. And we only analyzed the last 800 milliseconds before target contact. That's about the time frame of most good EEG experiments. So that's what we chose to do. Okay. Now, again, we're looking for tau. Tau is going to be the domain within which we look for neural resonance or neural coupling. So we can calculate tau for the ball, right? That is, as the ball approaches the target, it's got a tau value because it's a gap closing. We calculated the tau for the movement, that is the hand motion, same thing. It's a reaching motion, so you've got a gap closing there, so the time to contact. And then we calculate tau for each electrode. What does that mean? Well, remember that tau is just position over velocity. And so in the case of EEG, it's voltage over the rate of change of the voltage, right? So tau is just the thing you're looking at by the dot product, right? By its first derivative, right? So you can do that with lots of things, including the brain. And we did this because now everything is in the same sort of, as it were, mathematical domain of tau. So we calculated tau for each of these things. And again, to remind you, what we're looking for is tau coupling for the ball versus the hand versus the electrode, and the ball versus the electrode. All right. Now, for us, we wanted to make this hard for ourselves. So we picked a very strict definition for what tau coupling would be. And in our case, it's defined as a Pearson's R, so a correlation of 0.98 or greater between the time series. So basically, we're just looking for linear coupling. Again, the equation I showed you before, chi equals a kappa psi, right? So a linear coupling, but of such a strength, so it's a 0.98 strength. And as soon as it hits 0.98, we say that the two variables we're looking at are tau coupled. We did 10-fold cross-validation for each electrode. So it's, in other words, a leave 10% out fold process. And then the data that I'll present you are the average percent of time in the time series, that is the last 800 milliseconds, that the two variables are tau coupled, right? So what we're looking at is a percent of time, the percent of that 800 milliseconds in which we observe tau coupling. Okay. To give you just one example, this is not how I'm going to present the data in general. Let me get my thing back here. So this dotted line, that's the ball, right? And because it's moving in a constant velocity towards its target, you just get a nice, simple, smooth slope. Okay. The blue line is the hand movement. And what you'll see here is that it starts off not coupled clearly at all, but by the end of the time series, it coincides almost exactly, right, with the tau of the moving ball, which of course is what you'd expect if your task is to catch the thing. Okay, but nevertheless, it's a nice result because, again, I won't report on these data because I'm really interested in the neural coupling, the neural resonance. But the ball and the hand are over 60% tau coupled across all conditions, right? So for at least 60% of the time, you get a near perfect match, again, greater than a 0.98 correlation between the hand movement and the ball movement. That is not surprising. These are just two randomly selected electrodes in the facet medium condition. And you can see they have something like the same pattern here, whereas they're not really coupled at the beginning, but they become highly correlated near the very end, right? So as the hand is reaching the ball, the electrodes do become coupled. So I'll show you the real data here. You probably can't read this, but again, just so you know, right. So this is the percent of time. The red things are error bars. So the error bar coming from, this is the standard error coming from the cross-validation, right? So we're mostly interested in things with high tau coupling, so high percentage, but also low error. Because so some of these things that are huge error bars, that's probably not really a reliable coupling, right? So that's why we did the cross-validation. Anyway, we also did a permutation analysis because there isn't a direct way to figure out what's a statistical amount of tau coupling here, a statistically significant amount of tau coupling here. So we did a permutation analysis where we just mixed up the time series, and then we saw how tau coupled they were. And as you can see from the permutation analysis here, as soon as you get to 25% tau coupling, you're well less than point p equals, less than 0.01, right? In fact, you get to 0.05 somewhere here. So around 18 to 20%, you get to p less than 0.05. So anything that I'm going to show you is well less than p less than 0.01, 0.001. Some things way the heck out here, right? So this is, anyway, so just to let you know that we did do some statistical work here. So here is the fast condition and the coupling between the ball and the electrode. And these are just a list of the electrodes, right? So I'll show you these data on the electrode map, but just to give you an idea of what we're looking at, right? So electrode P05 is 46% tau coupled. C5 is somewhere between 30 and 60% tau coupled, right? Depending on how we want to deal with these error bars. You could probably ignore FC5 here, right? There's just too much noise to worry about it. But FT7, right? Very low error, and so it's consistently 40% tau coupled, right? And so on with the rest of these. It's more fun to look at it on the cap. Oh, well, sorry, this is just the fast electrode and hand movement. And what's interesting about this is we've got a whole bunch of stuff that's 100% tau coupled. So it's tau coupled for all 800 milliseconds that we analyzed. The other thing to notice, and we'll see this even more clearly when we look at the electrode cap and how this works, over time, right? So this is a percentage of time. So time is moving forward in the reverse direction, if that makes sense, right? So we're getting deeper and deeper into the motion as we get down here. But over time, more and more of the brain is tau coupled, right? So we're getting a massive spread of evidence for tau coupling across the surface of the brain, okay, which I think is kind of a cool result. This is the same with now we're looking at the electrodes versus the ball. And again, we have about 100% tau coupling for a couple of these electrodes, and very reliable too, right? There's basically no error there. And then, again, the same picture. You get increased tau coupling across the brain over time, right? As a task goes on, you get more and more tau coupling. All right. So this is a more fun way to look at it. So this is this electrode cap that I showed you. This is at time 0, so this is before the onset of the stimulation. And I'll show you at time 1, this is the hand movement and the electrode. At 800 milliseconds, we see all these electrodes being tau coupled, right, to the hand movement. At time 2, all the ones that were previously there are still coupled. Now we get more that are coupled. At time 3, oh, by the way, all our participants were right-handed, which starts to explain why you get more tau coupling on the left side of the brain. Right, but anyway, here's at 400 milliseconds to contact, and here's what it looks like at 800 milliseconds, or 200 milliseconds to contact, excuse me, right? So you can, I think, pretty clearly see massive amounts of tau coupling, right? And more as the task comes to its conclusion. Okay, that's the hand electrode. Here's the ball on the electrode. That's the visual information, right? So we have the tau of the ball approaching the target compared to the each electrode. So at time 0, of course, there's no coupling yet. There's also no coupling in this between these two variables at 800 milliseconds prior to contact. There's still no coupling at 600 milliseconds prior to contact. Now at 400 milliseconds prior to contact, now we start getting coupling between the visual information and the electrode. Now, if you've done any sort of visual neuroscience, this result may look surprising, right? Why that timing? Why that location of tau coupling? But again, keep in mind what it is we're measuring here. You would expect, if what we were looking for is the mere activation by visual stimulus, that you'd get very early activation in the back of the head. But that's not what we're looking at here. right? We're not looking at mere activation by stimulus. We're looking at the coupling in the context of a focused directed action. And here, I think what we see makes a lot more sense. It presumably has something to do with things like that's about when the participant starts actually paying attention to the location of the ball, because that's when it's looking, um, that's when it's approaching the target and where you have to really pay attention to catch it. No doubt, if we were looking merely for the activation by a stimulus, we would see early activity in primary and then secondary, et cetera, visual cortex, right? That's not what we're looking for here, right? We're looking for actual coupling between these informational variables. Okay, let's go back to the thing. So this is time 0, and I'll just skip straight to time 3. So this is 400 milliseconds before stimulus onset, before the capture, excuse me. And this is 200 milliseconds before capture. Now you see much more widespread tau coupling. And not much in canonical visual areas. But again, this is not really what we'd expect, because canonical visual areas were discovered and described in the context of looking for activation by simple stimuli. That's not what we're doing here. Okay, let me just show you these side by side. So this is the hand and electrode coupling. Hand electrode coupling here and ball and electrode coupling here. And as we saw already, at 800 milliseconds, we already have quite a bit of coupling of the hand and electrode. And now we have more. And now we start seeing coupling between the ball and the electrode. And notice that there's actually isn't very much overlap between these two. This once again underscores the functional differentiation here, right? So we'll still see functional differentiation, right? In fact, there's no overlap in the which are tau coupled at this juncture in the experiment. And here's 200 milliseconds prior to onset, and now we see some overlap, but not huge amounts of overlap, right? We're still getting differential tau coupling across parts of the brain, which I think is interesting and also kind of important. Okay, so initial summary of those results, I want to say, look, there's pretty strong evidence. Remember, this is R of 0.98. There's pretty strong evidence for very widespread neural resonance, right? Informational coupling between information in the environment, in the organism environment system, and neural activity. The coupling is only partially overlapping, as I just said, and it becomes increasingly overlapping as time to contact approaches. And of course, a less strict statistical measure would reveal even greater coupling. But I think, just to go back here, that's pretty good evidence for strong neural resonance, right, across large amounts of the brain in an environmentally valid task, even if we had to measure it in things like VR and EEG, right? Okay. So those are the data that we've just finished, maybe 2 months ago now, 6 weeks ago, we finished that analysis. So there's lots on our to-do list. Right now, we're working on what you might call a talons analysis. If you've read the neural reuse book, that stands for Translally Assembled Local Neural Subsystems. But basically, it's a kind of connectivity or coherence analysis. We're working on that now. We're debating whether we want to do a multiple dipole analysis. Right? Dipole basically tries to imagine what the sources of the electrical activity observed at the surface of the scalp is. I have my quibbles with dipole analyses, so I'm not sure we're going to do that, but it's certainly an option here. And then, of course, we still have to think this is the first time anyone's tried to do this. Anyone's tried to look at coupling across in EEG, across the brain, across behavior, right? So there's been lots of work done on the visual, and there's been certainly lots of work done on a tau analysis of behavior. This is the first to try to do behavior, visual information, and neural dynamics all at the same time. So there's certainly work we want to do, just making sure we have done everything properly. So that's on our to-do list. But let me just now summarize things a bit. So some important things to notice in what we're up to here. First of all, the task is not the passive viewing of a passive stimulus like faces. It's not the passive viewing of an active stimulus like movies. It's not passive viewing plus active thinking like, hey, add or subtract these two numbers. It's not the search for a feature or the count on an event, right? It's not all these things that have been contrived to work in cognitive psychology labs. Instead, it's a closed loop experimental paradigm. By closed loop, I mean it's one in which perception causes action, and the action causes changes in perception over and over again in a loop, in a fairly ecologically valid context, right? Just catching a ball. And this Davidson I'm referring to, that's Emily Davidson, who was an advisee of mine, got her master's degree. She's the one who did the Pac-Man study. And that's also a closed loop experimental paradigm, right? Where you play Pac-Man in the scanner. So what you're doing actually changes what you're seeing, and so on and so forth. As I mentioned already, I think we got some really nice results there. It was methodologically extremely challenging, mostly because participants got really engaged and excited, and so they wanted to move around a lot inside the scanner. And of course, as you know, if you know anything about fMRI, movement really messes with your results. So that was a challenge, but we showed it could be done. So hopefully people will be following up on that in the future. Anyway, more things to notice. The dependent variable is not the correlation of activity with stimulus, right? So it's not a general linear model approach. And the DV is not the detection of information allowing for the classification of an experimental event, like MVPA and RSA does, right? I do love those two methods as a nice innovation in looking at the brain in an informational space, but they're still generally pretty passive, right? Um, uh, what it is instead is the identification of an extended psycho-neural-behavioral coupling during a coherent, intentional, perceptually guided action. Okay, so here are my summative claims. So I think this approach may offer a better purchase on the causal involvement of brain parts over the GLM-based localization on the one hand, and over information-based analyses on the other, because we have evidence not just for the activation of these things by stimuli, or the ability to predict events based on information analysis. We've actually got causal coupling between regions of the brain, in fact, lots of regions of the brain, and the ongoing activity in behavior. So that's a very different thing to have discovered. I think it's of theoretical interest in its alliance with dynamic systems theory over, say, computational theory of mind. And I think it's of philosophical interest because it captures the brain-body environment system without the assumption of neural modules or mental representations, right? Nothing in what we looked at involved the representation of the ball, right? What we had was the coupling of the activity of the brain with the movement of the ball, the coupling of the activity of the brain with the movement of the hand, the coupling, of course, of the information specifying the ball movement and the information specifying the hand movement, right? So we have with causal coupling without any talk of or need for the talk of representations, and certainly no talk of modules, right? Nothing that I showed you in those pictures looked anything like a traditional neural module. Okay, in other words, the goal is less about functional mapping than understanding the parameters and constraints that govern the behavior of the system over time. And here are the references I promised you. So let me just, so yeah, here, this Davidson one, the 22 human neuroimaging reveals the agency in a video game. That's the Pac-Man study. That's right, that's the one. And then the two Rata ones. So Minds and Machines was indeed the 2018, and then 2021 was his Synthesa article. If you're interested in this stuff, I highly recommend taking a look at those. He and I have done some work together on constraint. So that's one you see there. And then a really preliminary analysis of the data that I just showed you is here in Raha and Graman in psychophysiology, just came out a couple weeks ago. And then just some other things that I mentioned along the way. I'm happy to show any of those slides again, but that's it for me for right now.
Thank you very much, Michael, for this rich and stimulating presentation. I'm also, working on some version of radical embodiment, and as a field, we have been getting the criticism that radically embodied approaches cannot make sense of neuroimaging data. To some extent, was right, but I think that is changing with some novel and creative works conducted by you and some other researchers as well. So thank you again. It was quite interesting for me as well. Um, we can now open the floor for questions and discussion. If you'd like to ask something, please click the raise hand button in the Zoom reaction menu. I will call on you in order. When it's your turn, you can unmute yourself and ask a question, or you can also type a question in the chat whenever you want. I will be following those as well. So we look forward to hearing your thoughts. I have a couple of questions. Maybe while others are pairing their questions, I can ask something. I mean, my first question will be about the these studies that you conducted about the functional diversity and categories and how they relate to each other in
Mhm.
a neuroscientific setting. Um, those concepts are actually, in a sense, folk psychological concepts, and there are some recent works that criticize those concepts, those categories may not even mean anything particular,
Yeah.
like Chisak's study, or I remember Gibson rejected the concept of memory altogether
Yes.
at one point. So, I mean, what do you think? If we could come up with a novel ontology, novel functional ontology, uh, would it could it say something different about the diversity, functional diversity,
Yeah,
or
yeah,
paradigm in general?
yeah, absolutely, it might. I wouldn't say they're folk psychological concepts for the most part, because a lot of the concepts they may have originated that way, but they've really grown up in the computational theory of mind, right? So they're really ways of operationalizing what may have started as psychological concepts, like emotion or memory, but then putting them into an information processing framework. Um, so they became, I think, scientific, at least to a large degree. Uh, so you presumably know Russ Poldrack has written about this, and one of his really nice articles, um, was it Functional Neuroimaging 2.0? It was one of the something from that era. Anyway, but he points out that, or he suggests that the reason we see the overlaps and the functional diversity, all the stuff that I reported on, so he refers to this work. He says, well, but the reason we're seeing that is because our ontology is wrong. So just as you wouldn't expect, say, phrenological concepts to map neatly onto the brain, you shouldn't expect these concepts to do so either. So we really need to get much more serious, in his view, about the computational modeling and get rid of the focal psychological content entirely, right? And then once we find the actual base operators in the computations, then we will start finding specialization of the sort we've been looking for a long time. I think that's very unlikely. And I think it's very unlikely for some of the biological reasons that I mentioned at the very outset. Not just my data, because my data are indeed contaminated by the very thing he says. If we have the wrong ontology, then my data are not telling us something. But if you see the same pattern of results in a soil nematode, you've really got to take that seriously, I think. Right? And you can't claim that somehow those behaviors aren't genuinely different, right? In the animal you're observing. So the Chisek stuff is also, I think, very interesting and very promising. But he doesn't expect localization either, right? He does think we ought to change our ontology and make it a much more behavioral and evolutionary infected ontology, and I agree with him entirely. But if you look at some of his most recent work, which I discussed in the book that I mentioned, um, uh, his way of discovering these behavior slash evolutionary um, uh, categories. Well, they don't map onto parts of the central nervous system. They map onto cooperative circuits across the nervous system that allow for the behavior to emerge, right? And yeah, that seems right to me. Paul and I have given a couple of talks together, basically making this very point.
Thank you for your answer. Yeah, I'm quite sympathetic to Chisak's work as well. In fact, I mean, I'm planning to work on something similar during PhD, but yeah, we'll see. Is there anybody else who wants to ask a question? I do have a couple of questions, but I mean, I don't want to kind of dominate the conversation. So, is there anyone else? Okay.
Either overwhelm them or I put them to sleep. We don't know.
Um, I'll ask another one. Um, about the, uh, ec- I mean, in traditional or normal cognitive science, um, ecological validity of experiments is quite problematic for too many reasons, especially in neuroscience, neuroimaging studies. Um, I mean, I was thinking of one reason for this might be the history of cognitive science has been dominated by experimental psychology and you know and neighboring methodologies. But when we look at the biology or ecology, there are some complementary ways to assess a given hypothesis. There are maybe I mean you might resort to some entirely observational methods as opposed to controlled experiments. Do you
Mhm.
think something like that is possible for cognitive science? I mean, like observational methodologies and experimental methodologies, uh, and can they be complementary in cognitive science as well?
Oh, I would certainly think so. Um, so for one example, now this is not a neural study, but I can imagine ways of incorporating neural, measurements into it. So I'm thinking of early work done, say, by Barker, which was almost entirely observational, really just documenting the behaviors of people in a town, right? It's fascinating work, and certainly has really important cognitive ramifications. Um, and uh, and then there's work, for instance, done on navigation, right? So that's also observation, right? How do people actually navigate from point A to point Z? Now, here with mobile EEG, you could absolutely incorporate neural metrics into a navigational study, which is highly observational. But you could look for, so I've looked at tau in a ball catching task, but you could look for tau, you could look for other ecological variables um, and constancies in a navigation task. I don't think there's any methodological reason that this wouldn't be a valid approach. And what the other thing, is a more comparative approach, and this is why Chizek's work is so important, right? A more comparative approach has to play a larger role, I think.
In fact, I didn't know that name. Can you put it in the chat?
Which one, Barker or?
No, not the last one that is
Oh, that was
quite special.
Paul Chizek again.
Oh, okay, sorry.
Yeah.
I thought you mentioned someone else that she said
Oh
got
well,
this
maybe
great.
I did. Maybe so Harry Heft is the one who did the
Oh,
navigation.
okay, yeah, okay, yeah, all right, okay. I know Harry. Um, okay. Doğukan, please go ahead.
Thank you for your presentation, Professor. My question would be about tractography. Right now, according to an article that I have written in Springer Nature, tractography has some accuracy issues, but if in future it could be fixed, what do you think it will like add to cognitive science? Could we see researchers about cognitive science where tractography is used?
No, so yeah, you mean like DTI and these methods for tracing white matter connectivity by tractography? I'm just going to assume that's what was meant. Um, so, so I've done some work with, we haven't published it because we weren't, for the reasons you sort of obliquely mentioned, some of the DTI techniques can be a little bit uncertain. But one of the things that Olaf Sporns and I worked on some years ago was comparing my functional connectivity analysis with his work in DTI, so the tractographic analysis, and the overlap between them is quite striking. So that doesn't mean connectivity is destiny, because we do know that there are changes we know there are changes at a very fast time scale in the connectivity between neurons. And we know that there's connectivity changes at a much longer time scale in connectivity across regions. So there's that level of dynamics that still exists. Nevertheless, the functional connectivity analyses that I did, that I showed you just a fraction of, and his DTI analyses marry up quite nicely, right? So that suggests, well, I mean, one thing it suggests is it's some of the spookier versions of neuroscience where you're really getting coupling across things, and I don't know, so the power band of EEG, or what have you. Some of the work in consciousness suggests this, that there's a binding problem and things are bound across the brain by um, by shared whatever 80 hertz oscillation or something. We don't need that kind of mechanism to make sense of behavior, um, because the physical connectivity helps us do it. Now, it's still not clear, given that we have the physical connectivity there, but we nevertheless see differential functional connectivity over time, how that's modulated, right? How do we actually control which bits of the brain are talking to which bits of the brain in a given time? And that part is interesting and not well understood yet. So I think the tractographic analysis is going to be really crucial going forward, but there are questions on top of even the reliability of the methods that haven't been answered, or even to my knowledge, much investigated.
Thank you. I think I have some kind of related question. Do you think the approach you just outlined here can explain or explain away the already piled up neuroimaging data? I mean, or are they just two different strands of research?
So I'm not sure I understood the question.
I mean, in neuroimaging, especially in using fMRI and other studies in cognitivist or some sort of computationalist approaches, in the history of cognitive neuroscience, we have been doing too many experiments, and there were like too many findings. So some person working in that school of thought might come here and say that, but how do you explain this phenomenon? How do you explain this finding, etc.? Do we have an answer for those kind of questions, or we should just say, okay, it's a different branch of research and this is another paradigm?
So, I mean, partly the reason I mowed the bulk of my work, not what I just showed you, but the bulk of my work has been meta-analyses. Because what I wanted to do is see what information was available in the data that had been collected already that could tell a different story about things like the overall functional organization of the brain. And indeed, you can, right? You can recover that structure, even though the data were collected under different assumptions. If you look at them the right way, then yeah, they can be mined to paint a different picture. So the data we've collected are not useless, even though they were collected under assumptions that are turning out to be fragile. So I think there's always opportunity to revisit existing data with a different set of assumptions and see if you can tell a story that militates against the prevailing one. And I think that ought to be possible, right? Unless you get sort of strict Kuhnian incommensurability, which I think most philosophers of science don't think there's that kind of Kuhnian incommensurability really, then you really ought to be able to largely reach back into existing collected data. I mean, that's the data were collected under circumstances that interpretation becomes impossible. But that's, I think, a rare case.
Thank you. In fact, I was expecting an answer similar to this, but I just wanted to make sure. There is another question. Nisa has asked it in Turkish, so I will translate it.
Yeah.
If this paradigm becomes the dominant view in cognitive neuroscience community, would that lead to change our research practices? For example, not focusing on fMRI, but focusing on other imaging studies? Or what do you think about this?
So I suspect we're never going to give up on fMRI, mostly because unless we come up with a very different technology that has the same strengths. Um, it's just way too powerful at imaging deep structures, which EEG can't really do, right? We're just looking at surface because of the deterioration of the electrical signal over time. Now, MEG can do it a little better, maybe. Controversy about that. But so I don't think we're going to give up on fMRI because of the strengths it does have. Now, it does have weaknesses, some of which I mentioned already. The one of I haven't mentioned is the temporal delay is significant. So there's some fancy math you have to do to kind of adjust for or even get rid of that temporal delay because the bold signal has that 2-second delay to it. I do, however, think, and this is why, the work I did with Emily Davidson and Jody Cullum is, I think, potentially important. We do have to start performing more ecologically valid experiments inside the scanner. And that's very difficult because, of course, the configuration of the scanner makes overt behavior difficult and disruptive of the data you're collecting. But again, we managed to get good, interesting data playing Pac-Man. And if we can do that, well, then why not a more realistic game like, I don't know, Halo, or pick your favorite, doesn't have to be a first-person shooter, but pick your favorite immersive video experience, right? Where you're actually acting in the world. If you can do it in Pac-Man, you ought to be able to do it with that. Again, there are still methodological challenges. For instance, deciding what events are, right? So what it is you're tracking. But so the stuff I just showed you, you don't necessarily have to be super precise about what the event is if you can identify what the ecologically valid information is, right? So yeah, I don't think fMRI is going away, but I certainly would like to see a much more concerted effort to make the experiments we run in fMRI much more ecologically valid.
Thank you. Is there another question? I think I don't have a question. Doğukan, please go ahead, please.
So now recently there are new programs, data mining AIs, which can analyze your data very efficiently for you. Are you using any of them, or are you planning to use any of them for this new paradigm you created?
I am not. I have some of my grad students are very interested in this. And in particular, this is more general than your specific question, but really trying to figure out how to use AI as an assistant in the scientific enterprise. So right now, it's really good at summarizing what we already know, and it's really bad at extrapolating. Right, that's where it makes its mistakes right now. So the question then becomes, how can you use it in a more extrapolatory manner, or to actually provide novel insights in structures you wouldn't have seen that turn out to be valid. Now, I mean, I haven't used AI per se. The techniques I've used have been machine learning techniques, and yeah, indeed, you can find patterns in data that are not obvious to human inspection. Here you face, and that's really cool and interesting. So back to Eunice, you mentioned the ontology. So some of the stuff I did using machine learning was to kind of reverse engineer an ontology given the structure of the data. And you can do it, right? You can discover structure in those data, but now you don't know what to make of them, right? So the interpretation problem becomes really acute. So no, I don't have, I did my postdoc in AI way back in the day. So I'm a bit mired in earlier technologies, but I certainly have grad students very interested in this. And I do think it's really important to be thinking about how to use AI in the way you describe, and to do it in a reliable way. So I don't think I'll be doing that, but hopefully somebody smarter than me will.
Thank you. Um, If there is another question, we can take maybe last 1 or 2 questions as well, and then maybe we can say goodbye. Seems like there isn't any questions. Okay, great. Thank you once again, Michael, for sharing your work and offering a thought-provoking, creative, and new ways to think about neuroscience and cognitive science in general. And thank you to everyone who joined us today. If you'd like to stay informed about our upcoming events, please don't forget to follow us on social media. And yeah, do you have anything to say, Michael?
No, it's just thanks for the opportunity. Hope you found it interesting and useful. And yeah, and if it was interesting and useful, hopefully you'll be amongst the people who follow up on it. Because I think there's a real opportunity to change the direction of neuroscience in a very positive way. So, but it's going to take the effort of many people over a long period of time. So, anyway, thanks for the opportunity, and we'll see you in some future meetings.
Thank you. Until next time, take care.
Right.
We hope to see you again. Yeah.
Bye-bye.